Hello, everyone. We'll just wait until another 1 or 2 minutes as people start joining the call. Thank you. Okay, I think we'll go ahead and get started as people slowly join. Hello, everyone. Welcome to today's MIDAS webinar. I'm Stephanie Shadbolt, the Outreach Coordinator for MIDAS, and I'm hosting the webinar today. For those of you who may be new to MIDAS, MIDAS is a global network of researchers, students, and others who are dedicated to the modeling of infectious disease dynamics. MIDAS membership is open to anyone who is working in or has an interest in this area. And you can find a membership request form on our website at midasnetwork.us. Just so that you know, this webinar is being recorded and the video will be available later on the MIDAS website on our events page. We're also trying for the first time to live stream to YouTube. So welcome to any of our viewers there on YouTube. We will have time at the end of the presentation for questions. And you can use the Q&A or the chat window to ask questions. You can also use the raise hand function and we can unmute you if you'd like to ask questions directly and make it a bit more interactive too. So now I'm very honored to introduce today's speakers, Dr. Isabel Rodriguez-Barraquer and Dr. Saki Takahashi. Isabel is an assistant professor in the School of Medicine at UC San Francisco. Her research focuses on applying novel epidemiological and statistical methods to understand the dynamics of infectious diseases and inform control interventions. Isabel was recently awarded an R35 grant from NIGMS for her project titled Uncovering the Dynamics of Human Pathogens Using High Throughput Multiplexed Serological Tools. And even more recently, Isabel received one of the new initiative awards as part of our MIDAS COVID-19 Urgent Grant Program. Isabel and Saki will talk a little bit about that project today. Dr. Saki Takahashi is a postdoc in the Experimental and Population-Based Pathogen Investigation Center at UCSF. As a Schmidt Science Fellow in 2019, Saki combined big data computing with molecular and serological techniques to explore complex infectious disease dynamics for pathogens such as malaria. And Saki is also a co-principal investigator on the MIDAS New Initiatives Award with Isabel. So now I'm very happy to go ahead and ask Isabel and Saki to continue with their presentation. So let me just stop my screen here. So Isabel and Saki, please go ahead.
Hello, can you hear me?
Yes.
Okay, perfect. Thank you very much for the introduction, and thank you very much for the invitation to present today. So yeah, we're going to be talking about some work we've been doing since March, trying to do some sero-epidemiological modeling and contributing tools for sero-epidemiological modeling for COVID. Much of this work, as you will see, is work in progress, very much so. But we want to give you an idea of the directions where we're headed and what we have accomplished so far. Next slide. So just as an introduction, we know that even though surveillance systems collect data on the number of cases for different infectious diseases, case numbers are per se a misleading metric of the number of infections. And this is true for many infectious diseases, including SARS-CoV-2, where there's a high proportion of asymptomatic infections. And in the case of SARS-CoV-2, well, we know that the vast majority of infections are actually asymptomatic or very mild, right? But it's even more misleading when there's incomplete understanding of factors leading to symptomatic disease, which is very much the case of SARS-CoV-2, even though we've learned a lot over the past months. And it's even more misleading given the differences in testing and reporting practices that exist between countries, and that's true for any surveillance system, but over time as well in the case of SARS-CoV-2. Next slide. So this is just some figures I pulled down from our world in data to emphasize how hard it is to really make interpretations of what's going on from case reports. So the left panel shows the confirmed number of cases per million people for 3 countries, the UK, the US, and India, I guess since March of this year. And you see cases rising and going down and rising again. But then on the right, you have the number of tests per 1,000 people, right? And you see that in essentially these 3 countries, the number of tests has been essentially increasing, which is great, but complicates the interpretation of the epi curves or the number of cases curves on the left. And that's why, well, in general, in terms of trying to make inference about what proportion of people have been truly infected, maybe using harder outcomes like hospitalizations or deaths is a little bit more meaningful because it also depends on surveillance, but let's say less so than cases which are highly biased or can be very misleading because of the reporting practices. Next slide. So in general, serology is thought to be the gold standard to quantify infections in a population. And serology, you essentially go out, take a small sample generally of blood, and you measure antibodies, which can be a biomarker of exposure in the past. Many of these antibodies last forever after you develop them, so they can be a marker of infection in the past. So it can tell you who has been infected at any point in the past. And in theory, it can also measure who is protected, which of course has important implications in terms of defining how much of the population is still susceptible for infection. But we're not going to get too much into that because using antibodies as markers of protection requires knowing for a fact that response correlates with immunity and protection, and that's a whole different story. If you measure antibodies and quantify what proportion of the population has been infected, well, there you have a direct metric of cumulative exposure. So attack rates, which you can then investigate across strata to identify risk factors, who's getting infected more, who's getting infected less, but also get at key epidemiological parameters as for sure what's proportion of the infections are asymptomatic or the infection fatality rate. It gives you a denominator to calculate the infection fatality rate, which has been one of the most elusive or debated, I guess, quantities over the epidemic. And really, in terms of transmission modeling, it gives you a different level of identifiability of many of the parameters in the model including are not, because just trying to estimate the transmission parameters from case data is complicated because there's many combination of parameters that could potentially give you the same shapes, and not knowing the proportion asymptomatics is a limitation to really nail them down. Next slide. So, of course, it became evident that serology was going to be important, and then since the beginning of the epidemic, maybe since April, different groups started publishing results from serosurveys. This is just a snapshot from Serotracker, which is a fantastic resource that tracks different serosurveys that are conducted. And I did just a search a couple of days ago, and over 130 population-based studies have been conducted to date. And this is just like the top 6 based on size. So we see population-based studies and like, I guess, probabilistic samples from England with 100,000 people, from Russia, from Italy, from Spain, from India. So yeah, there's a lot of interest. Next slide. But all of these studies are generally conducted different assays, right? Which is, and simultaneously, we're constantly getting headlines like this that are from 2 days ago. So one paper, Declining Prevalence of Antibody Positivity, a community study of for almost 400,000, and this is from the UK. But then the next day in Science, robust neutralizing antibodies to SARS-CoV-2 infection persists for months, right? Like, so how do we reconcile this kind of headlines, and how do we interpret results from serosurveys correctly? Next slide. Serosurveys are promising, but this requires having appropriate serological tests and knowing how to interpret them. Next. So back in March, at the Experimental and Population-Based Pathogen Investigation Center, which I co-lead with Brian Greenhouse at UCSF, we met as a team and tried to, and had a discussion of where we could potentially contribute to the COVID response. And among other things, we do have quite a bit of expertise, and we've been working over the past years on developing tools for Sero-epidemiology with a focus on malaria. Brian has an R1 developing markers for recent exposure, sero markers for recent exposure in malaria, but also we've done lots of works for arboviruses as well, trying to develop methods to make inference about transmission dynamics from serological markers. So we decided that was where we would try to contribute, and in particular, we've started several projects trying to get at some methods development for Sero-epidemiology for SARS-CoV-2, also characterization of antibody responses, and some projects on serosurveillance. And essentially, that's what we're going to be talking about, like short vignettes of the work in progress in many of these areas. Next slide. So as an outline for the talk, we're going to first discuss, our talk is going to include both parts on more methods development, but also parts on primary data collection and actual serosurveillance that we're doing. So first, we're going to talk about a study that we did in a low prevalence setting in Bolinas, California, and how to really interpret data from serosurveys, data from low prevalence settings. Then we're going to discuss some work we've been doing trying to understand biases, whether seroprevalence estimates are inherently biased and how to correct for that. We're going to be talking a little bit about characterizing the longitudinal responses, antibody responses, using a cohort of people with documented infections. And at the end, towards the end, we're gonna switch gears a little bit and discuss an ongoing project on surveillance, on serosurveillance in the San Francisco Bay Area, which leads to our MIDAS urgent grant. So Saki, if you want to continue from here. Sorry, and of course, if there's any pressing questions, feel free to ask through any of the Q&As on the chat, and we can try to respond as we go or at the end, depending on the question. Yeah.
Okay, excellent. Can you hear me? Okay. All right. So yeah, thanks, Isabel, and thank you for the introduction to present. So as Isabel said, I'll first be talking about a study in our community, in our local area in Northern California that Isabel and I were both involved in early on in the pandemic. And so this was a community-wide and community-initiated testing study conducted in Bolinas, California in April. So Bolinas is a rural town with a high proportion of elderly residents, with almost 50% aged 65 and older, according to the 2018 ACS, and has a population size under 2,000 people. And cross-sectional PCR testing and antibody testing was performed. And for antibody testing, we used two different serological platforms, an in-house IgG ELISA to the RBD, and the Abbott ARCHITECT assay. And in this study, there was over 80% community ascertainment, and there were no reported cases of COVID prior to the study in Bolinas. And for the PCR testing, 0 participants were PCR positive. And so this was a setting where we expected to see low seroprevalence or low prevalence. And so in this table here is just the raw antibody testing results for each of the assays, so Abbott and ELISA. And we can see that of the almost 2,000 participants with antibody test results, 8 individuals were positive on just ARCHITECT or the Abbott assay, 3 positive on the ELISA, and then 1 individual who tested positive on both. So just to refresh everybody, so I'm going to talk about test performance characteristics. So to understand the results of any assay, we need to understand how it performs. And so the way we quantify that, of course, is through sensitivity and specificity. And so to estimate the sensitivity and the specificity of an assay, we need samples from individuals with known disease status. So individuals who we know are infected, or for individuals that we know are not infected, as well as their results on that test. So whether or not they were positive or negative. And so, so basically we have positive controls who inform the sensitivity and negative controls who inform the specificity of an assay. And so if you do a serosurvey, you go out into the population of interest with your test, and then you measure the proportion of that population that you measure that tests positive. And so I'm just going to call that P observed. And then when we want to estimate the adjusted seroprevalence, you can take your test performance characteristics, sensitivity and specificity, as well as your observed proportion, and then do the simple statistical adjustment to get at your adjusted seroprevalence. And so these are the data that we use to validate our 2 serological assays. So for the Abbott test, this is data from the manufacturer on specificity and sensitivity. And so Abbott is, of course, a commercial assay. And then we also validated our in-house ELISA using positive and negative controls. Our positive controls were primarily non-hospitalized patients from a cohort study that I'll talk about later in this presentation. And so here again, we have our negative controls and our positive controls that we use to estimate test performance. And so just rearranging that simple equation that I showed on a couple slides ago, we can estimate seroprevalence given our sensitivity and specificity. So here, as a first pass, we calculated seroprevalence using each test separately. So here we have a binomial distribution to model the number of positive tests on test J, where N is the number of tests that were done. And we have a assay-specific sensitivity and an assay-specific specificity. And then we also have our assay-specific seroprevalence. And so we can make adjustments for the sensitivity and the specificity, assuming those are also binomially distributed. And for this study, we were also quite interested in the PPV, positive predictive value, or the probability of being diseased given that you test positive. And as I'm sure we're all well aware, tests that have high specificity can still have low positive predictive value, or PPV, when you're in a low prevalence setting, such as where we were here. And we also know that the specificity of a single test is likely not perfect. And so in this study, we had additional information because we had the test result of each individual on each of the two tests. So I'm just calling them tests J and K. And the test results I'm showing here with the positive and the negative subscripts for each of the tests. And so we assumed that the tests were conditionally independent, which means that conditional on the disease status, the test performance of one test doesn't depend on the result on the other. And so what we did was simply to estimate seroprevalence using multiple tests, we extended our binomial model from the single test scenario that I showed on the previous slide and used a multinomial distribution for the observed results. And then, as we've shown here, we can then calculate the two test positive predictive values. So PPV plus plus is the PPV for testing positive on both. Here, positive on one, negative on the other, and vice versa. And so I'm gonna just jump into the results of this study. So perhaps not surprisingly, our estimates of seroprevalence were quite low, extremely low. And you can see here that when we used each assay in isolation, our credible intervals for seroprevalence are quite wide compared to when we had more information and can use results from both tests. That allows us to get slightly better precision in our seroprevalence estimates. We can also see here that the specificity of each assay is quite high, which is good. But given the extremely low seroprevalence estimates, the positive predictive value of a single test alone, which is shown here in red, so for Abbott and then for ELISA, is not very informative. So the positive predictive value, which is this 44 and 63 values, are not very, they're quite low, and there's also a lot of uncertainty about these estimates. But when we bring in the results from two tests and analyze them jointly, we can see that there's high positive predictive value of both tests being positive and quite low probability of being positive, or sorry, quite low probability of having been truly exposed if individuals tested just positive on one test alone. And so to just quickly wrap up this part, our main findings were that 4 weeks into shelter-in-place health orders, active and prior SARS-CoV-2 infection in a rural Northern California community was quite rare. And from a more methodological standpoint, in low prevalence settings, we found that using 2 antibody tests can increase the precision of seroprevalence estimates, as well as our positive predictive values. Okay, so now I'm going to switch gears and share something that we've been thinking about a lot, which is about how the individual level antibody response to SARS-CoV-2 can affect downstream population level sero-epidemiological data and modeling. So as we all know, SARS-CoV-2 can cause a wide range of symptom severities, including asymptomatic infection. And there's a growing body of evidence that suggests that antibody responses differ by symptom severity. So here, I'm just showing 2 such studies. First here on the left, comparing antibody responses in mild cases to severe cases. And you can see that severe cases tend to have higher antibody responses. And this is another such study comparing antibody responses on a different assay between non-ICU patients and patients in the ICU. So we can see that antibody responses tend to differ by symptoms. We've also all seen various reports identifying and analyzing how antibody responses can wane over time. And so here, this is a report that was in New England Journal titled Rapid Decay of Anti-SARS-CoV-2 Antibodies in Persons with Mild COVID. So putting this all together, the question that we've been thinking about is if seroprevalence estimates for SARS-CoV-2 could be biased. And so for the reason that we've been asking this is that, so in the general population, we observe a wide spectrum of disease severities. And these, we know that these lead to differential antibody responses depending on symptom severity. We also know that antibody responses wane over time after infection. And then there could be an array of other factors that affect antibody responses, including age as well as immunocompromised status. But in the positive control sets that are used for assay validation, these are often limited to early convalescent samples from hospitalized patients with severe disease, and likely the individuals who have the highest antibody responses. And so in these validation sets, this tends to lead to very high performance characteristics. So here I've just pulled the reported sensitivity and specificity of the Abbott ARCHITECT assay, which you'll hear us talk about again later on. But you can see that there's quite high sensitivity and on this assay here. And so this discordance between the antibody responses in the general population and the antibody responses in the positive controls that are used for assay validation can lead to something called spectrum bias in epidemiology. And basically what this is when the assay sensitivity that you're using in the validation set doesn't reflect assay sensitivity in the general population. So to look at the potential effect of spectrum bias, we did some simulations. We simulated 3 different sets of positive controls shown here in 1, 2, 3 on this left panel. And we simulated what would happen if you were to validate the assay with these positive control samples that have different proportions of asymptomatic, mild, and severe infections. And then we simulated different values of true prevalence here shown in panel B on the x-axis. And importantly, we assumed values of symptom-dependent sensitivity, where individuals who had experienced severe infection had the highest sensitivity down to the lowest sensitivity in individuals who had an asymptomatic infection. And so this is for one test. And assuming these values of symptom-dependent sensitivity, we looked at the difference between the true prevalence in the population, shown here on the X, and the prevalence that we would estimate in the population, shown here on the Y-axis, using each control set, shown in colors. And we assumed that the true distribution of clinical outcomes in the population looks like this. So the leftmost bar here, back in panel A. And so, so the purple line here in panel B is where you use assay validation controls, which is set 1, that looks just like the distribution of severity in your general population in terms of clinical outcomes. And so here you don't get any seroprevalence biases or estimates, biases in your seroprevalence estimates. But otherwise, if your validation controls are skewed towards individuals who have higher antibody responses, so like in sets 2 and 3, then you'll underestimate prevalence. And so panel C, we just did some sensitivity analyses looking at how the extent of this underestimate would shift depending on what the actual values of symptom-dependent sensitivity are, since these are all simulations. And then we did some similar simulations looking at the effects of antibody waning after infection and how this could also affect seroprevalence estimation. And so we just simulated a simple epidemic curve shown here in panel A. And then we simulated 3 different serosurveys at different points along the epidemic curve. So at day 60, 180, and 300. And then for each serosurvey date, which is shown here in panel B, we calculated the distribution of time since infection at that serosurvey. So in this case, at day 300, many of the individuals will have been infected longer ago. So the colors reflect time since infection. And then we quantified the bias that would occur if The controls that were used for assay validation, which is the right column here, were all from recently seroconverted infections who have the highest antibody responses. And then lastly, here in panel C, we simulated crude values of waning, of antibody waning, using a very simple step function. And you can see again here that you'll tend to underestimate seroprevalence if you have antibody waning that isn't properly accounted for in assay validation. And in the real life situation, you'll probably have both severity and temporal qualities to think about. And so we also looked at what would happen if you compounded time since infection with symptom severity, and you see that the issues do compound. So based on these simulations, our recommendations to reduce potential spectrum biases were as follows. So first, we need to have longitudinal antibody kinetics in individuals who experienced a range of symptom severities, as well as other host factors by which antibody responses may vary. And ideally, this would be used for any assay used for serosurveillance. And then second, we really need a set of standards for assay validation, including, again, a spectrum of severity and time since infection. And lastly, we also think that we need more robust reporting criteria for assays, including the characteristics of validation samples. Okay. So as I just mentioned, in order to really understand the potential extent of spectrum bias, we need good information on longitudinal antibody kinetics across a range of severity and other characteristics. So now I'll share our efforts to do exactly this through a post-infection cohort study being conducted here at UCSF. So the title of the study is the Long-Term Impact of Infection with Novel Coronavirus, or LIINC study. So again, it's based at UCSF. And the study is enrolling and following individuals with confirmed SARS-CoV-2 infection from their early recovery period up to 2 years. And so as you can see from all of the photos, it's an enormous effort featuring many investigators. And while our team is focused on the antibody responses of these participants, folks are studying many different aspects, including immune cells and inflammation and other things using data from this valuable cohort. So individuals are enrolled during their early recovery period, which you can see here. And then they're followed up monthly for the first few months, and then every 4 months after that for 2 years. And to date, we have around 400 study visits in the first 6 months of the study. So we're around here right now where the yellow arrow is. And we have around 180 participants in this study. And so at each visit, various sample types are collected. And so the study is curating a really robust specimen repository, including serum, plasma, and PBMCs. And the study enrollment has had an increasing focus on enrolling certain populations, including individuals who've had asymptomatic infection. So these are the characteristics of the cohort to date. So you can see the age, this is the mean age, and the range ranging from 18 to 85. And then we also have information on sex, gender, ethnicity, and race. And in terms of the numbers here shown on the right, these are the numbers enrolled to date of asymptomatic individuals, symptomatic, and non-hospitalized individuals, which forms the bulk of this patient population, as well as symptomatic and hospitalized patients. And so here we're really excited to be able to have information on the asymptomatic individuals for this study. So like I said, our team's focus has been on measuring and understanding the longitudinal antibody responses of patients in this rich cohort. And so, as I'm sure we're all aware, the virus, the SARS-CoV-2 virus, has 4 structural proteins, including the spike and the RBD part of the spike, as well as the nuclear protein. And so we've generated data for each serological sample from a range of assays, and broadly characterized into in-house assays, including an RBD ELISA, and a bead-based Luminex, as well as some commercial assays, including the Abbott ARCHITECT, a diasorin in vitros, and then a pseudovirus neutralization assay as well from Monogram. So yeah, so these are the different target antigens on the different assays. So now I'm just going to
Saki, one short interruption, because this is something we haven't mentioned yet, but we are focusing this talk on much of the testing we've done so far on IgG, right? We've considered measuring other isotypes, and that's something we do plan to do, but for now these results are all focused on IgG responses in serum in particular.
Yes, yeah, thanks as well for the clarification. So I'm just gonna quickly share our preliminary results for 4 different assays. 3 are the chemiluminescent binding assays that are commercially available. So the Abbott, the diasorin, and the vitros IG, as well as 1 neutralizing assay. And so you can see here that the kinetics of antibody responses can be qualitatively different between the assays. And I should note here that the x-axis is days since onset of symptoms. And each line here is a participant, or each curve is a participant. And you can see that some SARS-CoV-2 antibody responses wane more quickly than others. So you can see here, the decay rate is quite high for the Abbott assay, and you have kind of qualitatively different characteristics for the other assays. And the dotted blue line here is the predetermined seropositivity cutoff as determined by the manufacturer. And so now I'll just highlight a couple different types of patients. So here is just one patient for whom on all 4 assays were generally of low responder. And you can actually see the seroreversion occurring on this diasorin assay for this patient. We also have patients who have robust and stable responses across the different assays. So this person shown in red. And then we also have other types of participants who exhibit more heterogeneity between these assays. So you can see here on the Abbott assay that this individual is approaching the cutoff for seronegativity. And so antibody kinetics have certainly been studied by other groups. And the rapid decay on the Abbott assay, which was on the top left of the previous panel, has been identified in other cohort studies as well. And so this is a figure from a group at Rockefeller University where they followed individuals who were mildly symptomatic and non-hospitalized, and they found similar decay of the Abbott assay as well. And so a key strength of the LIINC study is that the study has enrolled participants who've had a range of symptom severity. And so this is data from the LIINC study, again, on the Abbott assay, the one where we saw the waning, except now stratified by symptom severity. So our asymptomatic participants who were tested on this platform, symptomatic and not hospitalized, and then symptomatic and hospitalized patients shown here in red. And you can see that the asymptomatic infections or individuals tended to have low antibody responses on this assay. And then this mild or symptomatic and not hospitalized group kind of splits into these high responders and then low responders. And then we see that for the most severe infections, they tend to be high responders on this assay. And so as a first pass, we fit a simple hierarchical model of exponential decay to estimate half-life. And our point estimate was 101 days with credible interval of 88 to 116. All right, so this section that I'm going to talk about next is really the culmination of the last 2 sections that I presented. So, so we identified the issues that stem from spectrum bias. And then I talked about how in the LIINC study, we've been generating the data needed to quantify the bias. So now I'm going to bring this all together and discuss our ongoing work on how we can correct for this bias using a unified modeling framework. So just taking a step back, 2 of the large population serosurveys that have been done using this Abbott assay, where we see the waning, have been using this assay. So the large study in Italy and another one in Spain. And these are population-based serosurveys. And so we saw earlier that the magnitude of the antibody response depends on symptom severity and the rapid waning of responses. And so these are the raw seroprevalence estimates from these studies. So focusing in on the Italian serosurvey for a moment. So in particular in Italy, this is the epidemic curve of reported deaths. And as we all know, it peaked in late March. And then the serosurvey, which was done, was conducted almost 3 months after the epidemic peak. And so I just told you that there's an approximately 100-day half-life of responses on this particular assay. And so accurate estimates of seroprevalence are going to need to account for this timing, because antibody responses in individuals who were infected earlier ago, so you do a serosurvey here at this blue line, but antibody responses in individuals who were infected a couple months ago could have waned by the point of the survey. And so to obtain accurate estimates of seroprevalence, taking these things into consideration, we've come up with an analytical approach. So I'm just gonna quickly sketch that out here. And this is very much still ongoing work, but it's the exact same idea that I presented earlier using our simulations, except now we're fitting this to real data. So here, we're taking the time series of deaths and hospitalizations, focusing on these outcomes for reasons that Isabel described earlier. And then we're also taking our longitudinal immune response data specific to the assay of interest. And then we can use back calculation techniques to estimate the daily case onsets. And the need to do this is we want to calibrate all the data to be to the same metric of time. So here it's going to be time since symptom onset. And then we can use our kinetics data to estimate time varying assay sensitivity by different symptom severities. And then we can identify serosurveys of interest, so their date as well as the raw seroprevalence measures. And then we can put it all together into a Bayesian model framework to obtain adjusted seroprevalence estimates. And so seroprevalence estimates that account for time since infection as well as severity is going to be increasingly important moving forward. And this is because we'll have longer as well as more variable times since infection. So just to quickly summarize the last few sections, we know that there's a broad range in SARS-CoV-2 antibody responses that depend on symptoms as well as over time. And for serosurvey results to be unbiased, assay sensitivity needs to reflect the sensitivity of the assay in the population that you're interested in. And so we can leverage longitudinal cohort studies such as LIINC study to quantify key parameters for various assays. And this allows us to get at time varying as well as symptom specific test sensitivity, again, in our population of interest. And then lastly, we can use modeling frameworks to integrate these data types with information on the recency of the epidemic in a local area to obtain accurate seroprevalence estimates. So, so I guess before handing it back to Isabel, who's going to share about some of our other ongoing serology projects, we're happy to take any questions that you may have. But if not, I will hand it over to Isabel. I think you're muted.
Okay, so let's continue. So yeah, so now we're gonna change gears and talk a little bit about the serosurveillance effort that we have started in the San Francisco Bay Area. Next slide. So our thought back in March and April was like, okay, this we don't know how this is going to unfold. Seroprevalence data is going to become really important in order to know and properly identify where we are in the epidemic curve. So what do we do, right? Like, and we know that in general, the gold standard for sero-epidemiology are population-based surveys, where you go out and obtain a random or probabilistic sample of the population, right? But this is very expensive, for sure. It requires, well, getting teams out to the streets to sample a random subset of the population, and therefore, given the expense and the time, and how time-consuming it is, it's generally performed infrequently and in constrained geographic areas. So it doesn't really provide an information. It's very hard. I mean, I guess some countries have done it. The REACT study in the UK is sampling 100,000 people per month, but it's based on mail-ins, I guess. But in general, it's hard to obtain high resolution, temporally and spatial data using population-based surveys. And the fact is that to date, there's no citywide evaluation of seroprevalence in San Francisco. In contrast, there's thousands of people that are getting their blood drawn for clinical labs all the time. There's also people that are donating blood, and anyway, there's big numbers of serum samples that are collected routinely everywhere in the world, right? And it is true that many of these samples collected in hospitals are from sick people, but it's also true that maybe are not. And examples of these are prenatal visits, routine visits, or someone that shows up in the ER with a broken leg, perhaps, right? Maybe is not as biased, right? So our thought was that maybe with proper selection, right, this set of samples or this type of samples could be leveraged for efficient longitudinal serosurveillance at very high spatial and temporal resolutions that would otherwise be impossible. Next slide. So that's how we started a study we call Scale It, which is Serosurveillance for continuous actionable epidemiologic intelligence of transmission in the San Francisco Bay Area. And our model started with the UCSF hospital network and the county hospital, San Francisco, and Zuckerberg San Francisco General Hospital as well, so 4 hospitals. And this network collects over 50,000 blood draws per month, right? So what we thought in order to make a difference, because if you look at SeroTracker, you'll see that, I mean, there's plenty of studies that have been as well published now with this type of samples, but if we wanted to do something different is we really focused on thinking out how to do a good sample selection algorithm. And that's a big difference with, for example, the results that have been published by the CDC, which are based on big laboratory corporations, but they have no metadata whatsoever of the samples that they are using, or very, very little metadata other than location. So anyway, we included samples from individuals residing in San Francisco, or also including the homeless population, which accesses the county hospital. We excluded individuals who went to the hospital because of symptoms compatible with SARS-CoV-2. We did include some that were tested for routine purposes because the hospital started testing everyone that went into the hospital, but not those that were going into the hospital because of being suspect cases of COVID. We included both adults and children. In adults, we restricted to outpatient visits, but in children, we actually had to include as well inpatient visits because the children are not drawn blood as often, right? And as I said before, we focus on having like rich metadata, like in order to be able to weight our samples, in order to obtain a sample that was as representative as possible of the population in the San Francisco Bay Area. So we weighted by age group, and we weighted as well by zip code. And in parallel, we enriched for pregnant women as a true sentinel population. And also longitudinal samples, because those are nice to have. Next slide. No, go back a second. So the idea is that we get these samples, we biobank them in the lab, and we never imagined the amount of work that would be to collect up to 500 samples a week and biobank them and process them in the lab, perform laboratory assays, and it's taken us longer. I mean, setting this up took longer than we expected, but the original idea was to start doing this more or less in real time, processing them and having them in real time. Next slide. So yeah, so this was approved in mid-March. We started collecting samples in April. We haven't been able to test yet all of the samples that we've collected, but we have tested over 5,500 samples so far. And yeah, this has been like a huge effort in partnership with clinical and research services. And this has been truly a team effort from essentially everyone at the epicenter. And in terms of the serological test, we decided to do a two-stage sequential testing. First, like a screen with an ELISA against RBD. Again, we're measuring IgG here. And then, and particularly to improve specificity, a confirmatory testing using in-house LIINC assay against 3 antigens. And the figure on the bottom essentially shows the spread of the samples that we've been able to collect. So a very good representation of the city of San Francisco. On the left, a pretty good representation of age groups, less samples from children, unfortunately, than we would want, but in general, a good representation across age groups. And also a good representation, a good zip codes, even though we're working with the 4 hospitals shown in the figure on the left, like we are able to get with our weighting strategy, a sample from zip codes, like representing the true distribution of the population in zip codes in the city. Next slide. But I guess San Francisco has been lucky in the sense that the epidemic has been very limited so far. This is just showing epicurves of the cases and the deaths in San Francisco to this date. So there's been 12,000 cases. Lots of testing, but only 145 deaths in the city, which we're very fortunate about. But I mean, that also means that there hasn't been too much transmission. Next slide. So our preliminary estimates with the samples that we have processed is that more or less 4% of the population has been infected, and this is taking into account, these are preliminary estimates that take into account the assay sensitivity and specificity, like we've adjusted for them, how Saki explained before, to combine the 2 assays, I mean. But what's interesting is that this method does seem to capture the geographic heterogeneities, which are pretty striking. With much higher seroprevalence in some areas of the city, much higher seroprevalence in working populations and in Hispanic populations than in the rest of the city. Next slide. And of course, we are very interested in validating whether our estimates obtained with this Serosurveillance scheme resemble those from the general population. So UCSF conducted a community-based Serosurvey. I guess they also did PCR in the Mission District in late April. And this study, which essentially enrolled a very large proportion of all people residing in a census tract in the Mission, testing over 4,000 samples, found antibody prevalence of 3%, which was highly skewed towards workers. So much higher in essential workers, but overall 3%. So it's more or less in line to what we're finding in our residual sample study. Next slide. So we are really excited about the opportunities that this type of surveillance offers. This biobank could serve as continuous, this type of biobank could serve as a continuous serologic observatory for COVID, but also for other diseases of current or future interest. Of course, there's a trade-off between the potential biases of non-probabilistic samples, but then you have the advantage of a high spatial and temporal granularity of the samples. And here we're using a single hospital network, right? Like you could really expand it to include other hospitals to achieve much more granularity and arguably more representativity. Yeah. Next. So this nicely leads into the urgent grant that we were lucky to be awarded. Next. So we submitted a grant to MIDAS, and this is between me, Saki, Isabel Routledge, who's another postdoc in the lab. We are the three co-PIs and closely collaborating with Brian Greenhouse. And really the focus of this grant is bringing together all of the vast amount of information that all of these projects are generating. So with AIM-1, we want to formalize the frameworks that Saki described to really develop cross-scale analytical frameworks to accurately estimate SARS-CoV-2 seroprevalence and attack rates over time as space. So this includes kinetics models, and this includes integrating individual data on immune kinetics onto population models of seroprevalence in order to obtain temporally and spatially resolved attack rates at different scales. And the idea is to apply this to the scaled study, but also to apply this framework to other seroprevalence studies that are becoming available since we are lucky to have, and we will be lucky to have data on the kinetics of antibody responses for serial platforms. And the second aim is using the scaled data as well and integrating our spatially and temporally resolved attack rate estimates to develop mechanistic models rooted on Serosurveillance in order to assess like the dynamics of the epidemic in San Francisco. And here what we're gonna do is parameterize jointly using surveillance and serological data. And we're also we're collaborating with Google to incorporate Google mobility data, and Isabel Routledge is leading this part of the model and assess impact of non-pharmaceutical interventions. So that's the idea and what we'll be continue doing over the next 9 months. And I'm gonna finalize to finish with the acknowledgements. So next slide. Oh, sorry, I forgot the conclusion. So so serology provides, sorry about that.
Right, sorry.
There's a lag.
Isabel, I know we only have a couple of minutes left.
Okay,
so
yeah.
Perhaps, sorry, I know you've just reached the conclusions, but maybe we could just quickly address two of the questions that have come up in Q&A.
Sure.
So the question from Lloyd Chapman, what back calculation approach are you using to obtain the onset curve from the death curve? This is back when Saki was.
Yeah. Thanks, Lloyd. So certainly this is still in progress, and I would love to chat with you more about this, but we are exploring some of the, so there are some really nice packages that implement this. We've been looking at EpiNow2, which you might be familiar with, but basically thinking of various delay distributions that we would want to layer to go from the death curve to symptom onset.
Great, thank you. And there's also one other question from Alex Perkins.
So Alex is asking about integrating serological data to other types of data, and I think I briefly answered that in the last slide. So yes, that's definitely something we're planning to do as part of the MIDAS grant, is really integrating the different data streams in order to reconstruct transmission. Initially, our focus will be San Francisco, but hopefully generating methods that can be used in other places.
Excellent, thanks. And well, quickly, just one last question that's come in. Any comment on independence of serotests?
Yeah, I mean, I think that's a great question. I think actually that was a challenge that we had for the first analysis that Saki described, is that we didn't have joined the data, like let's say validation data or same sets of validation data run on the two tests. So we couldn't get at testing the independence. It is very likely that most of these tests, particularly the ones that target the same antigen, are not independent. But that's something that can be addressed directly analytically, as long as you have the right data, which is essentially the same set of control data on the different platforms. And that's exactly what we are trying to do for our analysis. Yeah.
Okay. All right. Well, we've reached the end of our time. So just wanted to thank Isabel and Saki very much for the excellent presentation. Lots of very great, impressive work, and look forward to seeing more in the future. So thanks very much again. And thanks to all our attendees. And the next couple of MIDAS webinars, we'll be hearing more from other grant recipients from the Urgent Grant Program. So thanks again, everybody.
All right, thanks. I think that went really well.